I'm Cindy Morris, a microbiologist from France's National Agricultural Research Institute in Avignon. In this short presentation, I'm going to talk about how some of the microorganisms that are commonly present on plants might have a role in rainfall that perhaps you had never heard about before. What are the factors that determine when, where, and how much it rains? First of all, the large-scale circulation patterns of the atmosphere create the conditions that make rainfall possible. But the atmosphere is full of particles called aerosols that are also involved in formation of clouds and rainfall. There are numerous natural and anthropogenic sources of aerosols that are regularly emitted into the atmosphere. Aerosols interact with ambient atmospheric conditions and can be decisive in whether rain will occur or not. Depending on their chemical or physical properties, aerosols have different effects on clouds and rain. There are three main types of aerosols that influence the formation of clouds and rain: cloud condensation nuclei, giant cloud condensation nuclei, and ice nucleating particles. Cloud condensation nuclei and giant cloud condensation nuclei catalyze the condensation of vapor to liquid cloud droplets. Ice nucleating particles catalyze freezing of cloud droplets to make drops that are heavy enough to fall as rain. Why is catalysis of freezing important for rainfall? You probably learned in introductory physics that water freezes at 0 degrees Celsius. But in fact, the spontaneous freezing temperature of pure water is -39 degrees C. At warmer temperatures, in other words between 0 and -39 degrees C, catalysts are needed. These catalysts are called ice nuclei. Therefore, in rain clouds, such as this cumulonimbus cloud, there are not always conditions where spontaneous freezing of water can occur. If the tops of these clouds are warmer than -39 degrees C, then freezing of cloud droplets will require catalysts. Freezing will be initiated in the cloud depending on the types and abundance of ice nucleating particles. In order for rain to form in such a cloud, ice particles catalyze the freezing of cloud droplets, then frozen droplets collide with supercooled liquid droplets, leading to the formation of drops that become heavy enough to fall. Other processes, such as collision of drops in very turbulent clouds, can also make raindrops form, but at mid and northern latitudes, freezing of cloud droplets is involved in most rainfall events. Other types of rain clouds, such as nimbostratus clouds, are not as tall as cumulonimbus clouds. Therefore, their minimum temperatures are about -10 degrees or warmer. For freezing to be set off in these types of clouds, ice nucleating particles that can catalyze ice at rather warm temperatures are needed. What is the nature of ice nucleating particles, and what is the range of temperatures at which they can catalyze freezing? This figure represents one of the most recent compilations of data on the freezing activity of the ice nucleating particles naturally found in the atmosphere. The x-axis indicates the temperature at which they can initiate freezing. The y-axis allows us to compare the rate of freezing caused by particles of different shapes, sizes, and nature. To compare the strength of the ice nucleation activity of all these different types of ice nucleating particles, the authors of the study calculated this force in terms of the number of ice nucleating sites per square centimeter surface area of each type of particle. That is what is indicated on the y-axis. Most of the known ice nucleating particles catalyze freezing only when water is cooled to below -15 degrees Celsius. These are the mineral and other inorganic particles that are very abundant in the atmosphere. Only certain biological particles, and in particular the bacterium Pseudomonas syringae, can catalyze freezing when water is warmer than -15 degrees Celsius. And furthermore, biological ice nucleating particles can incite freezing at temperatures as warm as -2 degrees. How do bacteria such as Pseudomonas syringae catalyze ice formation? Like all bacteria, Pseudomonas syringae has proteins in the outer membrane of its cell. One of these proteins has a special affinity for water. This ice nucleation protein binds water molecules, orients and organizes them near the surface of the bacterial cell. This makes the thermodynamic conditions more favorable for ice formation depending on the ambient temperature. This is a very rapid reaction, as you will see in this short film of Pseudomonas syringae inducing ice to form in water that has been cooled to -7 degrees Celsius. This water would have remained in the liquid state if we hadn't added the bacterial suspension. The main sources of biological ice nucleating particles in the environment are the microbial communities on plants. The outer surfaces of leaves, for example, are naturally colonized by bacteria and fungi in the same way that our skin has a natural microflora. Healthy leaves can have 10,000 or more bacteria per gram of leaf. Different species of plants harbor different quantities of ice nucleation active bacteria. Therefore, the amount of ice nucleation active bacteria that can be released into the atmosphere from plants depends on the species of plants in the landscape. In fact, most of the known ice nucleation active microorganisms are found on plants, in leaf litter, or in soil. Furthermore, the residues of these organisms, after they die and decompose, can adhere to mineral particles of the soil and maintain their ice nucleation activity. All of these different ice nucleation active microorganisms and the soil particles with debris from these organisms can be wafted up into the atmosphere. These different observations have led to the idea that ice nucleation active microorganisms are the key to a feedback cycle between land cover and the atmosphere. This feedback cycle has been called bioprecipitation. In this cycle, microorganisms are lifted into the atmosphere, they incite rainfall through their ice nucleation activity, and are deposited back to the ground with rainfall. The rainfall is favorable for growth of plants and microorganisms, thereby reinforcing the cycle. There is accumulating empirical support for bioprecipitation. Upward flux of ice nucleation active bacteria has been measured. Ice nucleation active bacteria have been found in clouds in several independent studies. In simulated clouds in experimental cloud chambers, ice nucleation active bacteria are able to cause the expected rates of ice formation. Ice nucleation active bacteria are in precipitation as it falls from clouds. The first strains of bacteria to fall with the onset of rain from a cloud are those able to catalyze freezing at the warmest temperatures. This is what you would expect if they were inciting the rain. Just after rainfall, ice nucleation active bacteria multiply rapidly. There is some evidence to indicate that they react more rapidly to rain than the rest of the microbial population on plants. And finally, the amounts of biological ice nucleating particles in the air just above plant canopies also increases with the onset of rain. This suggests that the generation of biological ice nucleating particles that can become aerosols is high when ambient conditions are propitious for rainfall. What are the implications of these observations for the policies and strategies we use to manage plants in agriculture, forestry, and agroforestry in particular? This is a critical question because land use is changing at unprecedented rates, with crops now accounting for at least 50% of vegetated surfaces and over 90% of managed land cover. If we consider the amounts of bacterial aerosols that are emitted from different types of land covers, it is clear that tree culture and other crops are responsible for the vast majority of microbial aerosols. Therefore, the choice of plant species and agronomic or forestry practices could have an important influence on the microorganisms on these plants. Furthermore, the geographic location of these plants could be important to assure that their microorganisms are released into the atmosphere in strategic contexts that have an impact on rainfall. Therefore, to manage crops and forests for their effect on the water cycle is not simply a question of managing plants. It is a question of knowing the microflora of plants and managing them in conjunction with the plants. To prepare this presentation, these are the sources of the images that I used. And if you are interested in obtaining more information, you can consult these links and request to join the BioIce mailing list.